So I'm Katie. It's really a pleasure to be here with you all. When I got the invitation to see, I was like, fantastic. I don't oftentimes get to talk about all the cell-fate transition work that our lab does. We talk a lot about synthetic biology. My training is actually, I'm a chemical engineer by training. And so that means I know very little about biology and about the same amount of math. So I'm very honored to be at a conference that is both math and biology. And our interest in the group is to, though, be able to use sort of the principles that do integrate between biology and math to build genetic control systems for the purpose of engineering cell therapies, and specifically designing genetic control systems that can function in primary cells. And so our testbed system is what I'll be telling you a lot about today, which is this primary cell system where we convert fibroblasts into neurons, and some of the things that we've learned along the way there. Again, it's really nice to be here. And I'm happy to talk about some of the work that we've done to basically ask the question, what is it that allows for cells to convert from one somatic identity to another, not going through an iPSC intermediate or a progenitor intermediate, and specifically asking how does cell state modify how cells interpret transcription factors. My group works on building genetic control systems and really thinking about how do we plug information into these systems, extract information out. And actually, we'd like to be able to do this in living cells. So a lot of the group works on the reprogramming side, converting these somatic cells, dermal fibroblasts from mice that we isolate from our mouse colony, or even human dermal fibroblasts. And thinking about then how do what are these gene networks doing? What are the cells doing generally? How do we interface controllers to perturb those processes to study them, but also ultimately we don't want to just do perturbation. We actually want closed loop control. And that's for the purposes of safety and efficacy. So then we're thinking about, well, okay, so if I want to make a certain perturbation, how do I actually design a genetic system that can actually enact that level of control? Thinking about timescales, the different molecular species, et cetera. How do you make sure you can actually achieve the result that you want, both through feedback and then also just inherent molecular biology? And so the goal is that we would actually be able to control gene expression, first of our controller, and ultimately of the cell. And if we think we can do that, we can probably control cell fate, although we'll also go for cell state. And we can have lively discussions about what those two things are. So the broad objective as an engineer is to be able to do this at scale for different applications. But I love that this meeting is really just about thinking about, how do we know what we know? And how do we know how transitions are happening? And this is a bit of a pure form of just like, whatever it works is good enough. I like that we're not thinking through just that. So although oftentimes I am defaulting to, is it good enough that it works? Because that's also important for some of the things we want to do. Okay, so for the cells we're thinking about, you have a fate 1, a fate 2, two different cell types. They're genetically identical to a first approximation, but obviously they look quite different. And so we have in our mind that we're going to then take and overexpress transcription factors, some set of them, and that will, along this axis, be able to move from one fate to another. And that's how reprogramming works. So if you're familiar with the Yamanaka factors, you overexpress this set of Yamanaka factors to make iPSCs. To do direct conversion, which is what I'm going to be telling you about today, we overexpress neuronal transcription factors in a cocktail that makes them much more powerful, and to be able to move along this line. But what we imagine here is that there's some sort of transfer function that's going to define how much we overexpress and how many neurons we get, or the probability at which a cell is going to transit to a new identity. And then the question is, how do cells interpret those levels? Are they the same all the time? And so this map, probably, there's people who've drawn diagrams like this all over the place. And it's easy. You actually just go into Illustrator, you open up a file, and then you can draw them. And so this is pretty straightforward. But these are mostly made by imagination, because we don't actually know how the different levels of transcription factors, especially at the protein level, the protein level, are going to map then to cell fate outcomes. That was the challenge we wanted to know. We wanted to know, how much more NGN2 do I need before I basically hit this jet band where adding more doesn't help. It just makes my cells more burdened. And so there's a practical application there, but also just how do these transcription factors work? This is what we wanted to answer. So I'll tell you a little bit more about the system and how we went about approaching that question, because it's actually really hard to do for a number of reasons. So this is a mouse from our mouse colony. You can see the HB9 GFP that's lighting up that spinal cord. That's how we read out whether the cells are actually motor neurons. Yes?
So the thing that always confused me when we thought about this a lot was the role of the medium
Oh, yeah.
the catching medium. So I'm just wondering, since you're trying to be quantitative, if you could maybe
we don't optimize or change the medium. We have a standard medium we use all the time. There's lots of different factors that go in there. We try, since we're overexpressing transcription factors, we have a basal condition where we don't have the transcription factors, so we can see what happens. So if you want to have comparisons, you can look there and just ask what is the impact of the transcription factors alone. But we don't. There's a few things we do for very specific perturbations where we're changing media, but it's asking a very specific question. Otherwise, we have a condition where we add plus or minus transcription factors to compare. Roll for that, yeah. So here you can see that one of the cells has adopted a neuronal identity. You can see that through the morphology. The reporter is turned on both of those cells, and clearly one of them looks like a neuron, and one of them doesn't. But if you were to identify these by flow cytometry, you'd think they were both neurons. And so if you're going to do RNA sequencing on these and put them into a population, how would you differentiate them? Maybe if you did single cell, you could tell them apart. So this is a challenge. The other thing that was a challenge when we first started on this is what you don't see here, because it's a live image, is all the cells that didn't actually reprogram, which was a huge problem when we started. For iPSCs, low rates and low efficiency, it's not a problem. iPSCs keep dividing. Neurons are post-mitotic. If you get 10 neurons, you got 10 neurons. You can sell them on Etsy. You can't do anything else. And so, even single cell sequencing, once you wash them enough, you don't have 10 cells. Like, you have even less. So we needed to solve this problem, basically, of how do we get enough cells. So I was having a conversation at Culture at Harvard for this meeting. It's called Cell-Fate Conversions. And one of the guys who's does some of this reprogramming work to neurons, he said, just blast them with transcription factors. That's how this works. If it's not working, it's a brute force process. You're just not using enough brute force. And I'm like, okay. So we tried more force. It didn't work. And I'm going to tell you why it didn't work. But it was not just that simple. Okay, so we really want to ask the question again, how do these transcription factors influence conversion? And the problem with this is there is an inherently rare receptive cell state. This is the hypothesis we went in with, and it's reasonable, but we also, it's something to think through. If you have a rare population that's going to reprogram, then your ability to observe that in intersection with any sort of other variable, basically it's zero. You can't do any sort of ability to infer a correlation because it's too rare. I think of this as kind of like if you go into a huge conference room or something, and some guy walks in and his name is Brady, and that's all that happens that day. You're like, is that a rare event? Should I have expected that event? But if 100 guys walk in and their name's Brady, you realize it's a Tom Brady conference type of thing, and like the Boston Patriots or something are playing, right? It's like, oh, okay, that makes sense. It's a correlation. It's that time, etc. Otherwise, one guy, you don't know. So you need enough events. And this is what we were working with when we first started. We had like 50 cells in a 96 well, which is like 4% reprogramming. It's really, really crazy low. So how then can we basically say, okay, do the transcription factor levels promote or impede or do nothing to the probability that we're going to get a conversion event? Conversion's rare already. Have I made it less rare? I don't know. My error bars are as big as anything else I'm measuring. Yeah.
Maybe too technical a question, but the example before you talk of doing this, I thought was done with microRNAs, which seem to be more efficient at doing
They're
this.
not, but yeah.
They're not more efficient.
Some people
chose
so.
to do this in transcription factors.
Yeah. Yeah, we wanted to ask that transcription factors, they're developmentally relevant. There seems to be a hierarchy of them. We could also do this process with microRNAs, but they're not more efficient.
They're not more efficient. Okay.
No, not in our hands.
Okay.
Yeah. But
maybe we'll get to this. But like, so did you have not to ask a mechanistic question, but like, do you have a hypothesis for what the mechanistic source for this extremely low conversion rate might be?
Such a great question. We'll get there. Okay. I promise. Ultra fast cycling cells seem to be your privileged population. This is something that was discovered in iPSC conversion. So fast cycling cells tend to become iPSCs. iPSCs are fast cycling cells. So do fast cycling cells become fast cycling cells by being fast cycling cells? It's sort of like do tall people become tall by being tall? It's really hard to know. But we can ask the question, in going to a post-mitotic state, do cells transiently rapidly proliferate on their way to becoming post-mitotic? And then it's not just a metabolic match, it's actually a phenomena that is potentially contributing. And as a chemical engineer, I thought this was very likely because the one thing that rapid cell cycle does is it gives you rapid turnover of your proteome. The same way a stirred tank gets turned over faster, the faster you dilute it. And that's what cell cycle did. And we actually talked about that. Someone talked about that earlier. Sorry if I'm forgetting. Was it Brian, maybe? Yeah. Okay. So then we use these insights and 6 years to make this slide, figuring out a cocktail. This gets to the question about conditions. We did use a TGF-beta inhibitor, but we don't change that anymore. to then make increase the rate of conversion. Now we have a system that's actually efficient enough. We can start to ask and layer on different perturbations and see if those perturbations are actually moving us up or down. Okay. So that now, again, we've sort of broken through the stochastic barrier. We can start to overlay different transcription factor levels and ask, okay, if I make this cell have this amount of transcription factor, is it more or less likely to become a neuron? Okay. And actually, I guess this is the first time I put these movies in. So here, actually, on the right, this is the low efficiency process. And then on the right is the high efficiency process.
Sorry, what is red?
So red is just a nuclear label. And the green is actually, this is, what's that?
Not every cell seems to be.
Yeah, so not every cell got the red label. We're actually trying to sparsely label them.
It's only for tracking.
Is it just for tracking? Yeah, the red's just for tracking. But the point is, do you see any green cells on the right-hand side? No. So can you tell if the red even correlates? It's completely useless, right? So here we can actually start to see the green cells are overlaid with the red. This is actually just asking the question, could we track them? And it's still, I don't know if you guys can see this, but they move so darn fast. It's really hard to do this by microscopy and then to do the image segmentation, but we're working on that. Because we'd actually like to do some like clonal tracking. If you guys ask me about clonality, I can't answer your questions yet. We can't do it by microscopy. Maybe Yogesh can help us to do it with barcoding, but we're not there yet. And again, they move so fast. So now though, we dealt this cocktail, we now have high rates. We had one more problem before we could actually fully solve this, and it was actually minimizing the cocktail. When we first started in this paper, we increased the rates of conversion. We had 8 different transcription factors on 8 different viruses we were delivering. That's crazy. And so not surprisingly, we saw high variability across batches of our virus and of our maps. And so we thought if we could reduce the chaos of that cocktail, potentially we could have better ability to infer how perturbations in the cocktail itself would influence rates. Okay, so we reduced this substantially. So Nat basically was like, his motivation actually wasn't entirely in the beginning to reduce the variability. He said, I just don't want to make this many viruses. And so I said, okay, we'll drop out some transcription factors and see what happens. I think it's going to get worse because, of course, if you drop out a transcription factor that's making your cell a neuron, it's going to get worse. It didn't get worse. It got better. He used the 6 factors, and then he compared that to NIL, which are these 3 motor neuron specific factors. We eventually figured out that BRN2 is inhibitory. I won't have time to tell you about that, but it basically slows cell cycle. And so this drag on cell cycle is really, really a huge variable in getting cocktails that are effective and efficient. Okay. So then we went in and wanted to actually quantify rates. This is actually really important. The standard in the field, I would say, if we're doing this with iPSCs, is challenging because the cells keep dividing. For us with motor neurons, like if you have one motor neuron, it's like one conversion event. We can directly map quantitatively. I had one event because I have one neuron. And so then we can also say, based on the number of cells I seeded, I can then say how many events do I get. And that's how we calculate what we call yield. Another metric that's also relevant for characterizing is potentially purity. In other words, at the end, how many of the cells are actually neurons? And basically this cocktail that we developed in the minimal cocktail, it improved both of those metrics, but they're still kind of low. We've actually pushed all these things. We can get up to about 60% purity. There's different things we can do to optimize the transcription factor encoding, et cetera. But this is about where we were.
Can I ask a couple of questions on the metrics?
Absolutely, facts.
So this is total number of cells at the end?
Yeah, at the end. So yeah, there's yield.
Which die?
Pardon? Oh, cells definitely die.
So then this purity, so I guess my question is,
of the surviving cells.
The top one, since you're getting yields which are greater than 100%.
Oh yeah, they're dividing too.
They're dividing a lot. Right?
Absolutely, yeah.
Okay, so you don't have a clear, so if it says the purity is 15%, it's probably less than that because many of them died of the ones that you started with.
Yes, so the overall outcome of any given cell in our population is not conversion. Mm-hmm. So that's why we want to trace clonality. We want to know like how many founders do we have that actually make it? We don't know the answer to that.
And it has to be less than 15%, much less than 15%.
In this one, it's 15. We've got up to 60. So the thing is, our range is changing. So actually, it'd be interesting to try some of these different systems and ask how many founders. Is it the same? Is it just an expansion? We don't know. There's definitely death that's happening. Quantifying that is like without microscopy, very, very challenging. Yeah.
And building on Yogesh's question, have you karyotyped the motor neurons at the very end?
We have not karyotyped the motor neurons at the end. We have looked, and so the 2019 paper, we went in and actually showed that we were less likely to get binucleated motor neurons from this cocktail, which previous versions actually you ended up with binucleated neurons, suggesting that there is actually some genotoxic stress. Additionally, before this, the cocktail we developed that was highly efficient, we would also see a micronuclei and chromatin bridges. So this cocktail actually eliminates those features. Seemingly, one of the reasons is because it actually activates the activity of the topoisomerase enzymes and then actually allows the transcription factors to be effective without genotoxic effect. So I skipped that entirely, but seemingly they're not worse neurons. And then by electrophysiology and other things, they're actually quite good. But those are great questions, because I think the thing is, you're like, you made more, but maybe they're worse. I'm like, seemingly not. We just got lucky. It worked. Yeah.
I guess I have a related question, because one of the motivations for this was to avoid changes in methylation of the DNA, because
Oh, you mean the direct conversion is
yes,
it retains
conversion rather than going through iPSCs.
Yep.
Have you checked that this method?
We have not checked. We're going to be working with some folks to do that. To be honest, aging and retention of aging wasn't really our interest. We also kind of figure
I know, but it was such a striking result for the.
it was for the gauge group. Other people have questions.
Okay.
So
Do you believe in the methylation story?
I don't know. I mean,
we haven't answered it. We wanted to work with some people who are more interested in going down that path before we spent the time, because we were like, we're kind of, honestly, if you remove methylation, we're like, great, you rejuvenated them. If you keep it, great, you have an aging model. But we're kind of agnostic as to whether, no one had the passion to go do it.
it does the same as the microRNA method. That's basically, which
Yeah.
was advertised to
Retain.
retain the original DNA methylation.
All direct conversion is sold as this. I don't
Yeah,
know.
but that's why I was asking
Yeah.
whether you check that.
Yeah, we haven't checked. Yeah.
All right, so you're working primarily in MEFs as your starting
Yep.
population?
Primary mouse embryonic fibroblasts. Yes.
Okay, so they're relatively young cells and they're
These
not.
are isolated from E13 to E14.5 embryos.
And have you also been porting these protocols over to fibroblasts from older
Human,
Yeah.
so adult human. Yeah. So if you go to the end of this paper, it's all adult human fibroblasts.
Okay.
And we've actually surveyed across a couple different donors, different ages. We've taken some patients who have Duchenne's and seen similar things. The cocktail varies a little bit, but the efficiency boosts are the same. We haven't looked at the transcription factor level data that I'm going to show you. We just wanted to actually, that's a 35-day process to make those cells. We've gotten it out down to under 28, but it's still really long. Yeah.
Great. And then, sorry, a second unrelated question is stoichiometry versus individual transcription factor level control. How much do those two?
Can you also repeat the question for everyone else?
Oh, so the question is stoichiometry versus individual transcription factor control. Stoichiometry, I think some of the next slides will answer some of your questions, but if they don't, you come back to me. But the general answer is NGN2 is really important. The others kind of are players that need to be there. They don't seem like they're that important. And in the follow-up paper that Nat had, we actually did all the arrangements of all three transcription factors. So you have 6 different cassettes, and they had different efficiencies, but the main criteria that dictated whether they worked was basically NGN2. Okay, I'm not gonna have time to tell you all about this.
3 minutes.
So fast cycling cells reprogram at higher rates. You can see this here, and they have greater neuronal morphology. So we wanted to go and ask, okay, how does the expression of the transcription factors change in hyperproliferative cells? We'd imagine that if those cells reprogram, they'd have really high levels of the transcription factors, right? No. In fact, when you go and sort them out, it's actually the hyperproliferative cells have lower levels. And again, if you believe in dilution, that makes sense. Great. But that also means we have a problem. If we are conflating these two populations and measuring the levels of our transcription factors, we're looking at apples and oranges. Okay, so we wanted to then go make these different cassettes. And here we're looking at NGN2 with a T2A Ruby. This gives us a proxy for NGN2. It is not the exact levels of NGN2 because it's a T2A. It's not the same protein. It just tells us they were co-translated. NGN2 will have its own separate set of regulations. So that's the caveat on this data. And then also we did this for ISL1, LHX3, and then we keep the other factors all constant. And by constant, I mean we deliver viruses. What we did here then was also, we were taking these viruses, we're putting them to MEFs, and then we do what we call a twin assay. So we infect the cells, we have on one plate, and we make a measurement at day 4 and ask the question, what are the levels of NGN2? And we've also made these constructs, so these UORFs give us different levels of expression. So we can have this axis of variation, and we can measure that at day 4. From that, we then measure at on our second twin plate. A lot of this was inspired by some of the work that Gesh did, and some of the other folks from Arjun's group. We take the other plate and measure how many IMNs do we actually get out of that? How many neurons are we making? So that we have an exact match data set that controls for any batch variants, and then we can make a map. We can say, okay, that we can say transcription factor level here, and then conversion here. This is what I had at day 4. This is what I have at day 14. Okay, so what do we see? So for NGN2, what we saw is that in general, the transcription factor levels generally correlate with increasing rates. And also, if you don't have NGN2, you basically see no conversion. So this is great, except everything correlates. And so what if it's just like, yeah, it's like your ability to express a transgene is what dictates whether you can program. Not surprising. So I was really happy when Nat went in and did this experiment. And for ISL1 and LHX3, you can see that the correlations are different, meaning that not any transgene is going to have this positive correlation, suggesting that there is an NGN2 or transcription factor specific effect that we're observing here. What's really interesting is also this is isolated. We've isolated just the hyperproliferative cells to exclude those non-hyperproliferative cells, because remember, in the non-hyperproliferative cells, their levels are really high and might not be predictive because those cells have very low probability of reprogramming. So what if we look at those? If we look at those, the correlations look very weird. NGN2 still shows some positive correlation, but those gray lines basically tell you if you measure the wrong cells, you can't see correlations that might be more important. We've gone back and overexpressed NGN2, and we see this dosage response. It's really beautiful. The final thing I'll tell you really quickly, given that NGN2 is so important for conversion, we wanted to ask the question, is it just NGN2 levels, or is it proliferation, and how much does either one matter? So and our hypothesis was that hyperproliferative cells will respond at lower levels. So Nat he went and did isolated different cells with different levels of NGN2 based on their cherry expression, and then reflated them. And then you mapped out the reprogramming efficiency. And so you see low, medium, high for these non-hyperproliferative cells, so cells that haven't rapidly proliferated and gone through that cell state. What happens? And you can see the rates are low. And then they're gated for low levels of cherry, medium, etc. What you see is at high levels, not surprisingly, the hyperproliferative cells reprogram at higher rates with high NGN2. But what's really interesting is even the medium levels of NGN2 reprogram at higher rates than the cells that have not rapidly proliferated but have high levels of NGN2, telling you that proliferation is more important than just getting to a high level. And these results actually continue at low levels of NGN2. If you've rapidly proliferated, you are more responsive to NGN2. And so that's how we draw these different functions there, indicating that proliferation history sets the receptivity and sensitivity of these transcription factors. Cell state, as defined by proliferation history, is important for how cells are able to then interpret these transcription factors and their receptivity to them. I'm way out of time, so here we go. That's what I told you. There's lots of other things that can influence transcription factors, but it's not just blast them and you get more, because we tried that and it didn't work as well as taking the cells through this proliferative intermediate and then to high levels of NGN2. And then you can see the human motor neurons there. There's questions. Sorry, I'm gonna go back real quick. Questions. Clonality, we don't know. Trajectories, we don't know. Epigenetic changes, we sort of know, but not exactly. And there's, if you like that, there's more. If you didn't like that, there's still more. And there's some tools on the synthetic biology side that might be interesting. So with that, this work was really expertly led by Nat Wong, who's a terrific founding grad student in my lab. And thank you so much for your guys' questions. Happy to chat probably at the break, so I don't take any more of Michael's time.